Welcome, everyone. I am Greg Veltri. I am a technical application specialist for de novo software, and today we're going to talk about spectral flow cytometry analysis techniques for FCS Express. I want to thank you all for coming today. Let's go over some housekeeping things. There is a question and answer button that you can choose. If you have any questions while we're going through this, I will try to answer them. If I don't answer it immediately, I will definitely try to answer it as we get to the end of our talk. All right, for today's talk, we're going to go through an introduction to spectral. So if some of you have been using compensation as your way to correct fluorescent spillover, and you're not really familiar with spectral, I'll talk a little bit about that. We'll talk about the instruments and data that we can import into FCS Express for spectral analysis. And then we'll go through some examples. We'll start with an unmixed data file to kind of get you oriented with the data. So that means it's been unmixed by the instrument that you're working with. And then we'll do FCS Express unmixing, which uses a wizard. And then we'll talk briefly about virtual unmixing, which is a way to do unmixing on conventional flow cytometers that don't have spectral flow cytometry. All right, so let's get started. So here I have a diagram of a cell that is labeled with a fluorochrome/antibody, placed in suspension and run through a cytometer, and we have a 488 laser, so we can kind of guess that this is probably FTSE. So as it goes through the laser and gets interrogated, what is happening in a flow cytometer is that it's producing a Stokes shift, and that Stokes shift produces a wavelength. And that wavelength is measured by the mean fluorescent intensity, and you get a curve. Now, cells are on one or more of the fluorochromes, and each having its own spectral distribution. So here I have an example of a three-color or three antibodies that have been stained on this cell. And you can see, what's this say? This is FTSE, PE, and PER-CP. Now, in conventional flow cytometers, what you have are mirrors and filters that are used to select specific bands of the optical spectrum. So you'll hear FL1, FL2, FL3, or detector 1, detector 2, detector 3, etc. And what it's doing is it's looking at the light just as you see it right here. Now with compensation, you're subtracting all the light that's not in the filter, and you're only seeing what's in the channel. And then what we do with compensation is we're going to use a subtraction of the signal to the channel so that we have just green light in FL1, we will have just yellow light in FL2, and we have just kind of orange light in FL3. And that subtraction is then taken away. So what you're getting is just the detector light and a subtraction of anything else that is overlapping into it. Now, with spectral flow cytometry, you're using prisms or gratings and arrays of detectors that are used to collect a continuous high-resolution optical spectrum. So you're seeing basically all of the light almost. The number of detectors in the array is larger than kind of the number of labels, and no particular detector is usually dedicated to a measurement of any particular label. Though I know in different instruments they will say, oh, use this detector, use that detector. But what it's trying to do is get all the information about this. And then someone wrote us a question as we're talking. Okay, we'll talk about that later. And then what you're getting is the sum of the fluorescence from each of these cells. Okay. Now here we get all of the FTSE, all of the PE, and all of the PER-CP. There are several mathematical models that can be used to perform the spectral unmixing calculation. We use ordinary least square method. I think that's similar to what Cytec uses. I know Sony uses weighted least square method. Beckman Coulter just came out with theirs, and I think they're just using least squared method. So everyone is using their own for whatever reason. But with ordinary least square method and all these type of algorithms, what we're assuming is a linear contribution of unchanged reference spectral. So you have to have your single positive controls and your unknown. And in turn, you're able to calculate the estimated spectrum for each dye. So now you're able to see each of these spectral patterns that we're getting here. And that's what spectral flow cytometry is doing. Now, the spectral data that FCS Express is able to read is compatible with most instruments that create spectral data. So the Cytec instrument, the Aurora and Northern Lights, Sony's instrument, the ID 7000 and the S8 3800, and in Thermo's Zenith, we're able to look at also. Now, each of these instruments export their data differently. So here's an example of Cytec. They have a raw and an unmixed folder, but it's really important that you're able to know this. So when you come into FCS Express with spectral data, if you're going to use raw data and unmixed data together, just know where the folders are so that you're able to read this correctly. And I'm going to show you in a moment where this information is for the instrument data loading in our options. All right, so let's bring in FCS Express. So I just want to mention this with our talk today. We're not going to be going through any of the basics in the sense of how do I bring in data, how to do things. We're just going to deal with spectral and how to analyze the spectral data that comes into FCS Express. All right, so if this is a little different for some of you who are looking at this, I just want to let you also know this is our latest version. This is the 7.28.0030. And within our software, if you go into File and you come down here to Options and go to Data Loading, FCS File Options, Instrument Specific Settings, you'll see here that we have an array of different instruments that have instrument-specific settings to help bring in the data easily. And you'll see if we scroll down a little bit here. Here's the Cytec Spectral Flow, the ID 7000, the SA 3800. We have the Bigfoot, Zenith, etc. Now, what this does for you is one, it identifies that it's spectral, but here are some general options that you should be aware of. This allows you to say the keywords and how you want to name them, in the sense in your parameters, if you want it name and stain, stain name or stain and name, etc. And then as we scroll down here, a little bit further, you'll see that you can change this, how it brings in the scatter parameters. But one of the specific options are importing spectra of individual lasers as individual parameters. So if you want to see each of the individual parameters, you can do that. You can import them as full spectrum individual parameters by parameter names, parameter stains, or wavelengths. And this is the same on all the others also. And then you can hide individual detector parameters from the raw data. And in Cytec and other instruments, we have a merge rule where you're loading both the raw and the unmixed data, regardless to which you're selecting, etc. So these are really based on what you want to do with your data. So someone has just asked me, are we compatible with the new BD Flow instrument? We're working with BD right now. We just haven't been able to get that completed yet with them. So hopefully soon we'll have something like that. All right, so let's close that. All right, so let's start with a file that has been unmixed already. So that means it went through, let's say, Cytec Spectral Flow, and it's been unmixed. So I'm going to bring that file into here. And I am going to create some plots. So I can select multiple plots. So I'm going to do a density plot and hit the control key and do a spectral plot with it at the same time. And we'll click OK. All right, and let's just do some things here so that it's a little bit easier to kind of see things. Let's expand this like this. Put this here. Negative. Go. All right, so we're going to just do some basic stuff just to get some things seen first. So let's just do the scatter plot. So do forward scatter, side scatter area. And then let's draw a gate around our cells of interest. And then you can see it. I'll just put it like that so you can. A new feature on this particular version is if you double click on this gate, you will get a plot. So I just thought I'd throw that in there so that you can see that. And then let's draw on the CD3s. And let's put a gate around the CD3s. And then let's do 4s and 8s. And once we get this all set up, I promise you, we will then talk about the spectral. So let's do on our CD4s. Let's put this over here. And last but not least, we're going to put a CD127 versus 25. And then these are your T-regs if you're not familiar with them. So let's put a gate around this. We'll just call these T-regs. And then put a gate around this. Show you some stuff. Let's get rid of yellow. And T-regs. Okay. All right, so we have the spectral plot here. We've done all of the basic things which we usually do, which is let's identify the cells of interest. We could have done singlets and all that kind of stuff live dead. And then we go through different subsets and we have this plot right here. So what can we do with the spectral plot? So first, let's look at it very carefully. We have violet, blue, and red. So we have all three lasers showing here. You'll see that this is the FCS file and that there's no gate on this presently. Now, if I right click on this plot, I can go to format this plot. And in the format this plot, what I'm going to choose from here is specific options. So in the specific options, this is where we have things that we are working with on the spectral plot. First is the resolution, and then you have other specific options. So I like to start with interpolate values. I'm going to click on that first. And our default is 5. So what that's doing is it's connecting all of those. So if you don't have a lot of detectors and you have minimal detectors in there, this allows the lines to kind of interconnect with each other and kind of give that line there. The next thing is the resolution of this. So a nice resolution that I usually recommend is about 200. And that kind of allows you to see things a little bit easier. Now, I want to remind you all right now that this is on no gate. So there are a couple of ways that I can change the gate on this. One is I can drag a gate onto here. Another is to select the plot and to change in the gating tab, the gate. Or I can click right on where it says no gate, and I can go to the populations that I want to work with. So let's do Treg. Okay. So this is live. So if I move this, this will also move the spectrum at the same time. I could have multiple spectral plots and be able to look at multiple curves on different plots. Or I can do what probably people are thinking is can you do an overlay on this? And I can. And I can do an overlay, say, on the T-regs versus the non-T-regs. So to do this, it's the same file. So I'm going to drag this file onto here. I'm going to add it and click OK. All right. So right now it's red, but I'm going to show you what's going on. So what we're going to do is selecting this plot. We're going to go to the overlays. And you'll see that we have both of the overlay spectrums sitting here. So this first one is on my T-regs, and the overlay to show is showing all of the spectrum. So what I want to choose is either the average spectrum or the medium spectrum. So I'm going to choose the medium spectrum. And then I'm going to go to the second file, and I'm going to change that to non-T-regs. And I'm also going to change that to medium spectrum. So now I have both of those curves there. And then which one is which? So what I can then do is go to the legend and make it visible. And now I can see that information there, the donor 1 or donor 2, but I can also change those names if I wanted to in the overlays and make the text legend. So we're going to make this non-T-regs. Right, so now we can see that there if I can spell correctly. So there we go. And then over here, we'll put T-regs. And again. All right, so now this is all live. I can move these and see where they are. So what's nice about this is that if you were trying to kind of figure out where your gates should be, or you wanted to give a spectral representation of the cells. Now, we don't all do cells. Some of us do bacteria, some of us do phytoplankton and other things that give off different types of fluorescence. You would be able to show that on this curve. Now, another thing that we can do with this is we can do high dimensional data analysis off of different things, and we can also gate on these plots. So what I want to just show you really quickly is an example of kind of doing high dimensional data analysis with what we have right here. So let's just go to tools. We're going to go to transformation. And in the transformation, we're going to create a new pipeline. The pipeline allows us to put different types of functions and different things on here, such as AI and all these flow AI and all these other cleanup type of things that we have embedded into here. So what I'm going to do is I'm first going to say what I want to gate this off of. So I'm just going to use my CD3 positives. Right. And in the pipeline, this is what I was talking about. And we have different talks that get into all these different things also. We don't have an option called like autospectral, and can it be done with our script? Sorry, we don't do plugins like that. So Adriana, who asked that, sorry. What we have is in here. We have different downsamplings, and what I'm going to do is a UMAP today. I've done this before for this particular one, so I'm just changing this just to make something a little simpler and quicker for us. So I did that, and then let's insert a plot. So let's do this. And then let's bring the pipeline. So I'm going to apply the pipeline to here. And what did I do wrong? I made a mistake. Oh, and did I not select the parameters? I didn't select the parameters. That's my fault. So make sure you select the parameters when you do this. So let's do this. All right. All right, and now it's calculating, and I know this is calculating because you can see it there. And then let's scroll down to UMAP 1, and then let's scroll down to UMAP 2. Okay. This is on no gate, but we're going to change that in a moment. All right. So right now it's a density plot. What I can do, and let's just close this for a moment, I can go to Format. I can change this to a color dot plot. And what I'm going to do is I am going to create a gate on my spectral plot like I was talking about. So we have where to place it. It doesn't matter as long as it's on some portion of the line of that you're identifying. So we'll call this spectral non-Treg. All right, and then I can go here. And then I can see it here. And then if I go to the dot size, I can make it larger. And what I can do is I can then duplicate this. And then we could draw another one here. I'll just call it gate 1 for now. And let's just bring gate 1 here. And you can see where it's in different places. So you can do a lot of different things with the spectral data if you want to use it on FCS Express. This is just one idea on how to do this by looking at the spectral of two different things. I could done this on two different plots if I was looking, two different data files and done an overlay like this. You can do a bunch of different things to really dig in and kind of mine the data that you have here. Are these features also available in IVD version? Yes, they are. So what I'm doing here, you would, the IDV version has all of the permissions. So that's really the only thing that you would be doing with that. You would just have to make sure whoever's doing it has the proper permissions. Okay. So this is if the data was unmixed. So now what we're going to do next is how to unmix the data on FCS Express. So let's minimize this one. Let's create a new layout. All right. And what we're going to do is we're going to go to Tools. And then we're going to go to Compensation and Unmixing. And this is what we're going to be using. This is the wizard that I was talking about. So there's a plus sign. You want to use the pull down. So you can do either compensation, but obviously we're doing unmixing. So let's select unmixing. We can give this unmixing a name if we want to. Let's say we'll call this 14 colors. And we're going to go step by step. So step by step, we're in the automatic unmixing setup is to first bring in the data files that we want to work with. All right. So I have a bunch of files right here. And so here's an example. If you have raw and unmixed, because we're doing unmixing, all the data that we're working with right now is going to be raw. So we'll use our reference groups for raw, which is all the single positive controls, and the multicolor samples will be raw also. All right, so someone's asking me about autofluorescence. We'll talk about that in a moment. So what I'm going to do is I'm going to select all my single positive controls. You'll notice that I had a autofluorescence 1 unstained sample. I'm going to bring that in a moment. I first want to go through just the basics of this first. So I'm going to do open. Okay. So within here, when you bring it in, it will automatically have the data file name. It'll then have the label that it goes with it. You'll notice that it says negative control is unstained because the unstained was identified automatically, and that is my negative control. Now, you can change some of these. So if you're doing cells versus beads, you'll notice this all says cells. You can have another unstained as another negative control of beads. And what you would do is right here, you could have the pull down and you would say which one you were looking for. So you would have an unstained bead instead of unstained cell. If you don't have an unstained, you can use the self as a negative also. So you have that option too. And then we'll come to this in a moment, but I'll mention you have a negative control, but you can also then add an autofluorescence control plus the negative, right? So, and we'll get to that in a moment. We're going to first do this, and I'm going to show you something that you can do. So the second step is forward scatter versus side scatter. So this is what my scatter is going to be. I know there are some instruments that use a forward scatter violet detector that you want to use. You can then select from here and do that. The next step is what parameters to unmix. So we're going to be using all three lasers, and we're going to select that one. So what you're going to see is kind of my mouse kind of doing this little crazy thing, and all the tabs are going to develop down here, which are going to be each of the fluorochromes. Okay. So let's do this. Bring this down here. Errors and warnings. Sometimes you will get a yellow warning, meaning that the detector, there's more than one detector that's being seen by this. It's okay. If you wanted to change that, you can go to adjust parameters and change it there. But if you get a red warning, then you should read this and see what the problem is. If it doesn't make sense to you, then just give us a shout out at support at denovosoftware.com and we'll get online with you and see what's going on with your samples. So let's take a look at what we have here. So I'm going to pin this window over here to the side. Let's make this a little bit more readable. Let's do this. We'll get more real estate. All right. First is scatter. This is the scatter for the fluorescence, and this is the scatter for the unstained. So we're going to just stick with this for right now, and we're going to go through each tab. All right, our first tab is CD3, and what I'm looking for is to make sure that this is sitting on the positive peak. Now, some people say I've been recommended that it should be the half portion on the top portion. The most important thing about this is that, one, this marker is live, so you'll see that the spectral will move as I move this. All right. So what I'm looking for when I'm doing unmixing is I want to make sure that I have a nice curve so that the algorithm is able to see that. So let's go to CD14. Now, this is great because CD14 isn't going to be sitting in this lymphocyte population. And for those of you who are not familiar with cells like this is lymphocytes. These are monocytes and these are granulocyte neutrophils. The CD14 is going to sit more over there in the larger population up here. So what I'm going to do is I'm going to select this gate. I'm going to go to gating and I'm going to make it data specific. So that means that this gate will be specific to this data file. So we expand it like that. What you see here are the two autofluorescents, and then this is the signal that I want. This is the CD14 that I want the algorithm to identify. All right. Then we'll go to CD45. That looks fine. CD8. We can change it if we wanted to, but it looks good for now. This looks good too. Let's just continue down the road. IGD. Okay, so not very bright, but we need to move this because it is sitting down over there. So let's move it there and select this and move this over here. Right. So I know that I am correct because you can see that we've changed the spectrum and we're getting something that is very similar to what it should be. So this is C4 BYG 667, so it should be right there. So that's good. Okay. Let's go to CD45RA. We could probably clean that up a little bit. Let's put the line like this. There we go. CD19. So let's do this. Clean it up. 25. Okay, so 25. We know that it is a smaller population, so let's make this smaller and bring it over. There we go. 127, CD16, 56, 4, 27, and then we have the unstained. So this is good. So this is the unstained that I wanted to, where I want it to be, and we'll bring about another autofluorescent population there in a moment. But let's first talk about the next tabs. So we have a visualization mixing matrix. So this is really nice because this kind of shows us this kind of where the peaks are and how they're sitting with each other. What I really like about this, if you see any peaks sitting below this line here, that probably means you need to go back to that population and see if you gated it correctly. All right, and then the next two plots are the spillover matrix, and then we also have over here the cross stain matrix. So let me just minimize this for a second, and I want to talk about those two. So the spillover spreading matrix versus the cross stain index matrix, and we get this question a lot, and I used to get it a lot as a director in a lab, and I thought that it was really important that you guys understand what they are if you don't know what they are. So the spillover spreading matrix measures how much spread noise from one fluorochrome another affects another detector due to the spillover. So its purpose is to evaluate how much spread a fluorochrome causes in a detector versus another one. Now, the cross stain index matrix quantifies the relative impact of one fluorochrome on another in a normalized way. And that provides a normalized index of how much one fluorochrome affects the other one. All right. So in short, the practical kind of takeaway of this is that the SSM, when you want to see the absolute spread effect of your spillover of the detectors, and then you use the cross stain index when you want to do a comparative normalization view to decide which fluorochromes are compatible in the panel. And that's probably the one that a lot of people will think about using. And they go, well, which one should I use? That's the one you can use to look at it and see. And so together, by using this, the SSM tells you the raw problem, but like I said, the CSI tells you how severe it is related to the other stains. So how well is your panel working, right? So that's really what you're trying to figure out in these big kind of stainings that you're doing here. And if you select this, you can then go see the whole thing and look at the whole thing. Now, you know, these numbers are kind of small. I know it's red, but what you have to do is you're going to have to look at them and see how they're doing, right? And that's what we'll show about in a moment. So once I have this matrix here, I can save this matrix. So I'm going to use the button right here. And let me go to where I want to place it. So let's place it in the spectral folder here. And this is going to be 14 color. I'm going to save that. All right. Now, for your autofluorescence that we were talking, someone asked about. So I can also have an autofluorescence sample too. And what that means is that my unstained is going to be used to do the subtraction to find the spectral, but the autofluorescence is another spectral color that may cause problems in here. Now, this is human peripheral blood. You don't really see the difference, and I'm going to show you in a second why that is, but this is the best example I could have for today. So let's open that. Let's continue. All right, so it's thinking. All right, so here it is. It said negative control. So this is where I'm going to change it to an autofluorescence control. All right, and then here's an example of the warning I was talking about. And all I did was I took the unstained and I copied it, I duplicated it, and just gave it a new name of autofluorescence 1. But that also means that they're both in the same detector. So this is what I was talking about. And that'll work fine for today. So the autofluorescence then has its own tab. So this is the tab. This is the marker that we had placed it for the unstained. But what I'm going to do is I'm going to move it over here. Okay. Now, how do you know one autofluorescence is different than the other? And it's the spectral curve that's going to help you. So if I were to look over here and think about it, you know, it's basically the same, right? I got a little peak here and I got a peak here, but when I go here, they're accentuated, and that's because the intensity is higher. So just be aware of when you're looking at autofluorescent populations, what you're looking for are kind of different curve patterns, not the intensity difference. The intensity difference will be different for each one of these up here, but really it's the curve change. And I don't have any eosinophils, but eosinophils usually have a different spectral curve versus the rest of the peripheral blood that you would see here. Now, I'm going to now save this one. So let's rename this. And I'm going to call it 14 colors autofluorescence. And I'm going to save that. And let's go back to where we were. All right, so we have the 14 color. Let's call it 14 color AF and do save. All right. Now I have this layout with all of the unmixing that I've done. What I would do is I would save this layout just by going to File, Save As, and then I would then, let's go to Spectral, and I will have it laid out with link data. You could embed the data. It's going to make it larger, the file. It's up to you on how you do your data stuff, but this is the way I'm just going to do linked for today. And this is my unmixed 14 color. The reason I'm saving this is that if I need a reference in the future of how I did the unmixing, where things were sitting on the markers, or I had a bad file, but I don't want to redo everything, I just want to replace it, I save the layout and I'm able to go back to the compensation unmixing, auto unmixing setup and change the files there. So we'll save that. All right, so let's close that and let's bring in a new layout. Okay. Now, before I bring in my data file, I'm going to go to Tools, and I'm going to go to the Unmixing. Let's put this over here. And I'm going to use the folder right here. And right now you don't see it, but if I select File Type and go to Unmixing, I'll have these two here. So you can only pick one at a time, if you noticed. So we'll first open that. So you'll see I have 14 colors, and then I'm going to open up 14 colors autofluorescence. All right, so this could have been with cells, this could have been with beads. So you can have multiple matrices on here that allow you to do comparisons, right? So that's the kind of the nice thing about unmixing and seeing where things, or maybe you had a different clone and you want to see if that affects it somehow. You can have all of those unmixing matrices sitting right here. So what I'm going to do is I'm going to bring in a data file. And let's bring this here, create a plot. And we'll just do a density plot, and let's put this over here, just so we can talk about this for a second. And then we'll click make a forward scatter, side scatter. And we're not going to do anything fancy like before. This is more of just so that you can see a difference. We'll do gate 1. And let's see, we got CD3. Let's do CD8. CD3. See if we can send, yeah, there we go. Maybe something like that, so we can see the difference. Okay. All right. So what I can do is I can select the plot, and I can go to the matrix here, and I can go apply to the selected plot and apply it that way. And then I can also go here and apply it to all the plots. Right. So I have one way of doing it that way. Another option that I have is I can right click on this plot and go to Format and go to Overlays. And what you'll see is I have the compensation here. And what I can do is I can pick the one from here. So I can apply the 14 colors here. And then I can apply 14 colors autofluorescence there. And you'll see it says transformed. So you would have to change it and write it in there if you wanted to. But this allows you to see, and you can see there really isn't a difference between these two. Did you import the raw spectral data or the unmixed? This was the raw. It just happens to be, this is a really good panel that's been placed together, but we can go through here and see if we can find something else. And we can do none and see if it applies it. So it's just a good panel. But the most important thing is not so much that you're going to see a difference here, more that you're able to do the different matrices to allow you to look at different plots and different things. Now, let's say you don't want that. What you can do is in that compensation and layout, if you want to have a default, which is the one that works on there when you bring each of the data files in here, all you have to do is do set as default. And that will always apply that particular matrix to every data file that you bring into this. Okay. All right. The other thing I want to mention that you can do, let me copy this and we'll go to another page. Let me just paste this here. Is n by n's. So I can select this plot and I will get a multi-plot right up here. And then I can do multiple types of n by n's, but I can do comparisons of how well it did. So this is a lot. I'll click OK. Let's see how it does. Uh, I only have it up for 10 parameters, but we can change it to more in the options. But we're able to then look at each one of these, and we can also apply different matrices to this also. Okay, so that, in short, is how you would do the unmixing and then how you would apply different unmixings to the data that you have. Now, the other thing that I mentioned was virtual unmixing. So let me show you that. Okay, so I've already done this because it takes a little bit of time to do. But what I've done to do virtual unmixing is the first thing I have to do is go to Tools and go to Transformation. And I have to create a pipeline. And in that pipeline, in the miscellaneous, I'm going to do merge the spectra. What the merge to spectra does, it produces a file that you're able to save, and then that output then places it into a folder. And so let me get that folder so that you can see what I've done. And it creates this keywords. And what the keywords will do is it'll have all the parameters in there and all the names of the parameters so that you're able to create a virtual spectral plot of the detectors that you have in there. So once you have that in that folder, it has to be with the single positive controls. You can then create the unmixing. So you bring in the data files. You'll have to type in the labels for each one of these. And then you can go here and then you would do the same thing. Now you'll notice that the resolution is a little bit different than what we've been looking at. And that's because I haven't changed the options on this. So it's just looking at what fluorescents are in each of the individual detectors. But it puts it all together and it allows you to get a matrix together of that. So if you're doing 4 detectors versus 20 detectors, you're going to get a better spectral curve with the 20 detectors versus the 4 detectors. So that's just something to keep in mind. But this allows you to do, if you want to do the BD data on here on the FCS Express, all you have to do is virtual unmixing and do this. On our website, we have a video that goes step by step on this if this doesn't make sense to you. And also you can contact us at support. But this is one way to look at your data, spectral data, or your conventional data spectrally. All right. So I want to thank you for attending. That's our talk for today on spectral analysis with FCS Express. Um, we have a lot of different videos and different tutorials at denovosoftware.com. And then also, we always are available to help you at support at denovosoftware.com. We'll get online with you and try to answer your questions one-on-one so that we can see what your data looks like and see what's going on. This video will be available in 1 week. We will send out an email next week. This is Wednesday, so next Wednesday. That will have the link to it so that you're able to watch this. Or if you want to show colleagues this video to help you out, help them out too. And then I'll take any questions since we have a little bit of time.